While I'm sure that most of you wouldn't make the same mistake as my student in this cartoon would, okay, we are going to talk today about meiosis. Meiosis is, again, a form of cell division, and meiosis is an important type of cell division because it's through the process of meiosis that cells are able to produce offspring with the diversity needed for them to be successful in their environment. And so we'll start here with a simple definition of meiosis. You can use this one to compare it to the definition we had earlier of mitosis. And with mitosis, we only produced identical daughter cells. But as you'll see here, meiosis is a process by which not only do we create 4 cells, but those cells are not identical to the parent cell. Those cells have only half the number of chromosomes in each of the cells. Now again, this is due to the fact that meiosis's goal is to create cells that can be used for the process of sexual reproduction. Okay? As a result, as I wrote there in the notes, it does not occur in every single cell. Now, I want to discuss a few key terms that we'll use in this lesson. The first being the term homologous chromosomes. Homologous is an adjective that's used to mean identical. And so when we refer to homologous chromosomes, we're talking about chromosomes that look the same under the microscope, that basically have the same information on both chromosomes. And so it turns out, again, due to sexual reproduction playing out, that in humans we have 23 pairs of these homologous chromosomes. Okay? 23 pairs comes to a total of 46 total chromosomes. Okay? And so when we talk about these chromosomes being 23 pairs, you can see here in this chromosome spread that, again, all the chromosomes look alike. And so for chromosome 1, basically they have the same bands, same appearance, all the way down to chromosome 22 and etc. One of the things to understand about these chromosomes is that each chromosome pair one of the chromosomes came from your mother, and one of the chromosomes came from your father. And so your total 46 means that your mom gave you 23 of them, and your dad gave you 23 of them. And so the fact that they came from 2 different parents is part of the reason why you end up with this variety during sexual reproduction. Now, second 2 terms here, the terms haploid and the term diploid. The term haploid is a term that's used to describe any cell that contains 1/2 of the normal set of chromosomes. Usually the letter N is used for cells that are haploid. So in humans, a haploid cell would be N equals 23. Okay? A diploid cell is any cell that contains a full set of chromosomes, usually with the abbreviation 2N. So a typical cell in a human that had a full set of chromosomes would have 2N, or 2 times 23, or 46 chromosomes. And so these terms refer to the chromosome set that's found in each of these cells, or in these different types of cells. And again, most of our cells are diploid, although our sex cells are haploid. The next 2 terms we'll have here are the terms gamete and zygote. Gamete is a generic term that is used for any sex cell. And by saying sex cell, we also imply that therefore it's going to be haploid in nature. So in animals, the gametes are the egg or the sperm. And in humans, again, every egg would have 23 chromosomes and every sperm would have 23 chromosomes. Okay? In plants, again, an egg would be a haploid cell, but it's not called a sperm, it's called the pollen. The other term that we have here is the term zygote. And the zygote is any cell created when a sperm joins with an egg to create a fully functional cell, a diploid cell with a full set of chromosomes. All right, now let's talk about the steps of meiosis. Meiosis is named after the same steps that you see in mitosis, except they add a 1 or 2 to it. So prophase 1, okay, what happens here is, again, the nucleus basically disappears, okay, and what happens is the chromosomes that have replicated are now chromatids, and these pairs of chromatids form groups of 4 known as tetrads, okay? And so this is unique to meiosis, and it only occurs during prophase 1. And so once this has taken place, okay, the nucleus gone, these pairs of chromatids forming tetrads, okay, is completed, then prophase 1 is all finished. Now, one of the important things that occurs during prophase 1 is once these chromosomes, or correctly, let me say it, chromatids, are paired up, then what will happen is crossing over will occur. Crossing over is this process by which, okay, pieces of the DNA from these homologous chromosomes will switch places. This only occurs during prophase 1, and what this allows to happen is this allows the beginning of the possibility for the cells to increase their diversity. And so once these chromosomes are now matched up, and you'll notice in this diagram that illustrates this process of crossing over, you'll notice you've got the blue and the white chromosomes, okay? So then they replicate in that second group. And then, again, crossing over, what that does is it takes those 4 chromosomes, or should I say more correctly, those 2 pairs of chromosomes, and by switching places, now you've got 4 unique chromosomes. You still have a white one, you still have a blue one, but now you've got 2 chromosomes that have information from the blue and the white one recombined. This is an important aspect of the meiosis process because of its ability to increase that diversity that is so important for the organism's survival, and thus also one of the reasons for meiosis. Now, once prophase 1 finishes, the cell is ready to continue into metaphase 1. When the cell continues into metaphase 1, okay, just like in metaphase in mitosis, okay, the chromatids in this case are going to line up along that equator of the cell. Again, the chromatids are still going to be moved around to keep those tetrads intact. Spindle fibers will permit this movement to occur, and once the various chromatids / tetrads are lined up along that equator of the cell, then metaphase will have drawn to a close, okay? And you can see this in this next picture, which illustrates the fact that, again, those chromatids remain in these crossed over pairs. So they're still in groups of 4 chromosomes, okay? Now, with anaphase, 1, just like before. Now the chromosomes, or let me re-emphasize that the chromatids will begin to separate. What makes this, again, different from mitosis is we're keeping these chromatids intact. And so at this point, the chromatids will move to opposite poles of the cell, and again, being pulled by the spindle fibers. And once they move to the opposite poles of the cell, at that point, anaphase 1 will have drawn to a close. Now, once anaphase 1 draws to a close, the cell is now ready to bring prophase 1 to closure, which means telophase 1 will happen. During telophase 1, the cell is going to divide into 2 unique cells, but when the cell finally divides into these 2 cells, those cells are not going to be functioning at this point, because the cells are going to contain not individual chromosomes, but the cells will contain pairs of chromatids. And the fact that these pieces of DNA are still attached together and still linked together as chromosomes instead of being unraveled, and in the form of chromatin, does not permit the cell to use the DNA at this point. So again, the cell will divide, and you'll have 2 cells, but these 2 cells will not be functional at this point. And once this happens, telophase 1, and also as a result of that, prophase 1 will draw to a close. But it's important to understand that meiosis has not finished at this point. Now understand that the cell goes through a resting point and a resting stage between meiosis 1 and meiosis 2. And once that resting stage is done, it will enter the second part of meiosis, meiosis 2. Now we're going to begin meiosis 2. It again starts with prophase 2. And when prophase 2 begins, remember now, this is going to occur in 2 different cells simultaneously. And so as prophase 2 begins, chromatid pairs will be released from the nuclei. And so as each cell enters prophase 2, what will be visible will be chromatid pairs. Now, these chromatid pairs will eventually be attached to spindle fibers in order to allow the cell to begin the process, of manipulating these chromosomes and moving them, around within the cell. Now, once this happens, once these chromosomes are released, once the spindle fibers are attached to them, the cell will undergo metaphase 2. And again, the chromatids, the individual chromatids now, will move to the center of the cell, where they will align along the equator of the cell. And when this finally happens, then metaphase 2 will have drawn to a close. Now, with anaphase 2, at this point, you finally have individual chromosomes moved to opposite ends of the pole of the cell. And so only during this part of meiosis will individual chromosomes finally appear. But as a result of the two divisions, what will happen is towards each pole will move only a half a set of chromosomes. So in the cell that you can see in the drawings, we started out with 4 chromosomes in the cell. Now, during anaphase 2, 2 chromosomes will eventually move to each pole of the cell, again occurring in 2 cells simultaneously. Once this occurs in anaphase 2, then what will happen is telophase 2 will come into play, and with telophase 2, you will finally have, again, the cells begin to divide, and when those cells begin to divide, okay, you will end up with, finally, 4 haploid gametes. In other words, these 2 cells that have been going through meiosis 2 finally will each divide, and when each of those 2 cells divide, they will each have only a half a set of DNA. And when these cells are finally produced, at the end of telophase 2, you will have 4 gametes. Each gamete, okay, is possibly a functional gamete that can provide a half a set of DNA to permit the cell, okay, to serve as a means of carrying out sexual reproduction. Okay? And so this occurs finally at the end of telophase 2. You've got new nuclei in each cell, and once the 2 nuclei form, the cell will cut it, each of those 2 cells will cut themselves in half, and as they cut themselves in half, again, the 2 gametes will finally be created. All right, so to try and recap this again, meiosis requires 2 cycles of division in order to accomplish its goal of finally reducing the cells to having a haploid set of chromosomes in it. So as you look at this drawing, keep in mind that the goal of meiosis is different than mitosis. The goal is to get the chromosome number reduced to a half, and that requires carrying out 2 cycles of division. Because you'll notice, if you go back and look at these drawings, that at the end of meiosis 1, there's just 1, okay? At the end of meiosis 1, each of those 2 cells has a full set of DNA in it. By the time meiosis 2 has run its course, that full set of DNA has been reduced to a half a set. Now, that's one of the jobs or functions of meiosis. Its goal is to do that. Its goal is to reduce these gamete cells so that they only have a half a set of DNA in them. Why a half a set, you may ask? Well, you got to think in terms of what an organism needs in order to function. An organism needs, in order to function, a full set. If each organism only carried out mitosis, then let's assume your parents only had mitosis and your dad's sperm and your mom's egg had a full set of chromosomes in it. That means you would have a double set of chromosomes, 46 from mom and 46 from dad, and you'd have a set of chromosomes, 96 chromosomes. Let's say the second generation, you have 92 chromosomes, excuse me. You and your spouse each have 92. Your offspring would then have 188. So without this meiosis taking place, the number of chromosomes in each offspring would continue to double, okay? And so this process is important for that. All right, to finish up this lesson here, again, the second reason for meiosis is to provide this variability that we've talked about earlier. And it provides a means of allowing each parent to provide their DNA in order to ensure the success of their offspring. It allows for crossing over to occur to increase that variability, okay? And since only one of the 4 gametes is used, again, variability occurs because which one of those four is going to determine the genes or the traits that the offspring will have. And so the big picture here is that, okay, meiosis accomplishes the halving of the chromosomes, and 2, meiosis improves variability amongst the offspring because crossing over increases variety amongst the chromosomes that could be inherited. Each parent contributes a half of the DNA to their offspring, and the fact that 4 gametes are produced and we don't know which one of those 4 might be used, again, adds another layer of variability in there. And so if you look at this drawing, again, you can see how mitosis and meiosis are different. 2 cycles for meiosis, 1 cycle for mitosis. Bottom line, same chromosome number for mitosis, halving the chromosome number for meiosis, thus the 2 cycles. And so you need that.